The Galien Foundation fosters, recognizes, and rewards excellence in scientific innovation to improve the human condition.
André Malraux said once in one of his novels, "Une vie humaine ne vaut rien, mais rien ne vaut une vie humaine." Human life is worth nothing, but nothing is worth a human life. All these men and women are trying to save human lives. Is there anything more noble than that?
The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year Best Pharmaceutical Product, Best Biotechnology Product, Best Product for Orphan Rare Diseases, Best Medical Technology, Best Digital Health Solution, Best Incubator Accelerator Equity, Best Startup. Around the globe, the Prix Galien is considered as the equivalent of the Nobel Prize for the industry, mobilizing an unrivaled network of Nobel laureates and top biomedical scientists. The Galien Foundation manages an independent, cross-functional, and geographically diverse program of events and sponsorship to brand the best of the best in new medicines and diagnostics. The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Roy Vagelos Pro Bono Humanum Award for Global Health Equity is bestowed to an individual, a company, an academic institution, or a non-governmental agency that has helped to improve the human condition through the application of biopharmaceutical science to problems of developing or underserved populations worldwide.
This is the right event, on the right issue, at the right time.
Staging this event in Africa is a significant statement that science and innovation are needed as much here in Africa as anywhere else.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
Well, welcome everyone to the Galien Foundation Week of Biomedical Innovation webinars conducted from New York and around the world. Our objective this week is to provide additional content and background for our largest annual event, which is the Prix Galien USA Awards, to be held this Thursday in New York, in conjunction with the annual Galien Forum of speakers representing top scientific experts and leaders in public health. Because the mission of the Galien community is to improve the human condition, we've developed this webinar series to give our nominees across many categories: Best Pharmaceutical, Best Biotech, Best MedTech, Best Rare Orphan, Best Digital Solution, and Best Accelerator, Incubator, and Startup categories greater visibility in describing their journey of medical discovery. This year's Galien USA Awards has a record 176 nominees in 7 categories from 169 companies, covering 18 diverse therapeutic areas, ranging from cancer, cardiology, neurosciences, to infectious disease, and very rare genetic diseases and vaccines. We'll be looking closely at all these products across all the critical areas in the coming throughout the week. So in the Best Pharmaceutical category, which is our focus today, we're holding 5 webinars, given that there are a large number, 24 different medicines represented in this important field. The webinar today is devoted to the 3 nominees joining us in autoimmune, metabolic, and genetic diseases. Our discussion around these vital products will focus on the complex biomedical, environmental, and genetic interrelationships that characterize these diseases. How has research science progressed in ensuring patients with these complex conditions can benefit from more personalized precision approaches to treatment? There will be some time at the end for audience questions, so you should have a link provided on your screen where you can forward those to the moderator, me. There should also be a link that provides access to the 100-word statement submitted by each company on their nominated products that can give you, a bit more detailed information. Okay, so now let's move to our discussion. I'll start by introducing our three nominees who are joining us for this webcast. So first we have Francisco Leon, CSO and co-founder of Provention Bio, a Sanofi company for TZIELD. Thank you, Francisco. We have Mike Vincent, SVP and CSO of Inflammation and Immunology at Pfizer for Sabinco. Welcome, Mike. And we have Paulo Fontoura, SVP of Global Health of Neuroscience, Global Head of Neuroscience, at Roche for Ocrevus. So welcome to all of you. So my first question, and maybe I'll start with Francisco, is just talk a little bit about, why this condition? What was compelling about this indication in terms of its accessibility or importance to patients that drew you to it?
Thank you, Mike. Well, when Provention Bio, now a Sanofi company, decided to focus on type 1 diabetes or T1D, it was because of the unmet need. You are all familiar with the progress in technology. We had exogenous insulin, automated delivery devices, continuous glucose monitoring. But despite all of these advances, 75% of patients with T1D do not achieve glycemic control. And that leads to complications. That leads to cardiovascular disease. If you get diagnosed with T1D before the age of 10, you lose close to 20 years of your lifespan to the disease. So a huge unmet medical need. And that's what we wanted to focus on, to try to bring the first ever disease-modifying therapy to this devastating disease.
Thank you. Yeah, I'm sure we'll get more into the science of it, but thanks for the overview. Maybe Paulo, same question, why focus, where you did anything about obviously the severity of the disease, but also the state of the science that you want to highlight for us?
Yeah, thank you. So Ocrevus, we decided to develop it for MS, multiple sclerosis, which is still today it's the leading cause of neurological disability in young adults. And what we're trying to do really here was that up until Ocrevus, no drugs were really approved that robustly control disease progression, so the accumulation of disability. And that's really where the medical need was. So it wasn't like for type 1 diabetes where there were, several therapies. For MS, fortunately, patients have had many options for a number of years, but still the leading cause of disability was not being controlled. So the idea was really to try and take a fundamentally different scientific, approach to this by targeting B cells, B lymphocytes, which at the time when we started was really not thought of being important in the pathogenesis of this, disease. So in some ways, it was a huge scientific leap of faith as well. But then it turned out to be actually the key fundamental, if you want, mechanism to allow to control progression in these patients.
Great. And Mike, same question to you. Give us, a good understanding of why focus where.
Well, our drug Sabinco is approved for atopic dermatitis, and it so happened that we've been working for almost 25 years on a class of enzymes that inhibits a variety of inflammatory cytokine molecule signaling. And we made the observation that several of the cytokines that are thought to be key drivers of atopic dermatitis could be impacted by a JAK1 selective isoform. over that 25-year period, simultaneously an appreciation of the scale of the unmet need in atopic dermatitis was really evolving. There were no approved therapies in the first 15 of those years, and in large part, atopic dermatitis was considered as a disease of childhood that most people outgrew. But in fact, when you look at the statistics, there's really an enormous population, even into adulthood. 5 to 10% of people over 18 suffer from atopic dermatitis, and it's a very taxing disease in terms of one's quality of life. The itching is really an incredibly bothersome symptom that people can't go through their day-to-day existence without thinking about. And so it was really the fit between an emerging and appreciation of the unmet need in atopic dermatitis, as well as the attributes of a kinase inhibitor that we thought we could design that would address the critical need in this illness.
Yeah. Okay. Great. Thanks. So I think that the three of you have helped orient everyone to the space we're playing in. Maybe to go a click deeper on the science side. So immunology, probably one of, if not the most complex, disease areas or therapeutic areas where our clinical understanding of the diseases and maybe is orthogonal to the underlying mechanisms. It'd be great if you can. Maybe I'll start with Paulo. B cells, right? I mean, lupus, right? There's other indications where that plays even outside of think of B cell lymphoma. Think of even outside of metabolic disorders and into cancer, or immunology disorders into cancer. But kind of bring that to life for us. What is it like to work in the science of this space that is so complicated, where different mechanisms and different indications are almost orthogonal?
Yeah. So, I mean, in MS, researchers have been trying to find a causation for MS for over 80 years. It's going to be a century now. I mean, we've known about the disease for over a century. And it's a remarkably complex disease, right? When you look at the pathology, both in humans and in animals, you find, as you usually do in autoimmune diseases, really a mix of cells, of cell types, of cytokines. And therefore, in that very complex puzzle, it's hard to determine which one is the key linchpin, right? And so approaches originally were actually pretty broad in terms of immune suppression, just trying to control everything. It turns out that's not viable, and it's not very effective as well. And as years passed by and therapies were being, approved, we really were falling into this kind of conundrum of either having relatively low efficacy therapies that were very, very safe, or really high efficacy therapies, the big guns, that were really not safe at all. And then we're talking about a population of patients which are mainly diagnosed in their 20s and 30s, mainly women of childbearing age, and therefore the risks that you're willing to accept for that population are also very different. So I would be I would not be true to form if I would not say that for the longest time, myself included, everyone thought that MS was a T cell, a T lymphocyte driven disease. All the animal data pointed towards that. All the genetic data pointed towards that. It was really when we started doing, experiments with drugs that depleted B cells back in the early 2000s with rituximab that we found out that actually depleting B cells has a remarkably profound effect on disease, activity in these patients, whether measured by, imaging, MRI, or even clinical outcomes. And so the idea behind, Ocrevus was really to test that robustly. But as I mentioned, it was in some ways a little bit of a leap of faith, because obviously there was that evidence coming in from clinical trials, small-scale clinical trials, but the proof was not really there. And in some ways, we were flying against conventional wisdom in this disease. So when we actually saw the results at the end, it was a remarkable outcome for us, and for patients. And in some ways, it has created a whole new line of research around the role that B lymphocytes play in MS. Now, you're right. In other conditions like lupus, of course, they play a big role. What I think is really remarkable about multiple sclerosis is the fact to which B lymphocytes seem to be really master controllers of the pathophysiology. And that was not something that us or anyone was really, expecting to be so.
Yeah, thank you for bringing that to life. I mean, it's a great example of scientific discovery, right? There's a little bit of a, serendipity, right, in the initial observation, but obviously the skill to notice the observation and then to follow it up with a scientific program to prove or disprove the hypothesis. Mike, maybe the same question to you. I mean, Jack Stat, I mean, you could make the same kind of discussion. I mean, talk to us a little bit about that again, how you think about how the pathway plays and different indications and
I mean, in very general terms, I think we often learn a lot from genetics about what drivers of disease are, and what we don't learn from genetics, we learn through trial and error with therapies. So I think we understood reasonably well that the cytokines that are important in driving allergy are cytokines that are integral to the development of atopic dermatitis. One thing we weren't as aware of were that some of the same cytokines that are upregulated in allergic responses don't just act on immune cells and skin cells, but they can act on nervous system cells. So in particular, a cytokine known as IL-31 is really crucial to telling the nerve that you feel the sensation of itch. And because we knew that itch was such an important symptom and such a bothersome symptom for patients, the knowledge from previous therapies that Th2 cytokine inhibitors like IL-4 and IL-13 inhibitors would be somewhat effective, knowing that we were going to suppress those pathways well with a JAK1 inhibitor, and then over and above that, block this itch pathway, made a really compelling case that, were we to study the JAK1 class in this disease, we felt pretty confident we would see a beneficial effect. Another, many cytokines that JAK1 inhibits, and a variety of diseases in which JAK1 inhibitors are known to have some activity. This, I think, was just a fit with unmet need at the time. This new realization that we would be attacking one of the more profound symptoms that was not inhibited by existing therapies really led us towards atopic dermatitis as a place to begin.
Yeah, I mean, for me, it's a great example of follow the science, right? I mean, the immunology plays a lot. Like, we just heard a neuroscience example, and we've heard what a classically would a dermatology example, right? But obviously, that's the right way to think about it is what you both have done, right? Which is, well, where is this actually playing in terms of impacts for patients, and follow the science where it leads. Francisco, maybe a slightly different question to you, which is, as you thought about transitioning, especially at the time, and smaller biotech, transitioning from proof of concept to product to market, and helping patients, what was that journey like? What were some of the challenges you faced, and how did you manage it?
Yeah, it was complex because TCL teplizumab was developed in a slightly different fashion as the typical immune drug. Our goal was to attempt to prevent the onset of clinical disease by treating patients in pre-symptomatic stages. And that was difficult because First, it required understanding the natural course of the disease really well and the pathophysiology that's common to all of us. So once we understood there's a genetic predisposition, there are events that break immune tolerance, and then the autoimmune disease has metabolic consequences. Once we understood that in type 1 diabetes, there are autoreactive T cells that destroy the insulin-producing beta cells of the pancreas, then the question was, can we identify patients early enough to preserve those beta cells? Because otherwise they'll be lost, and once they're gone, the pancreas doesn't regenerate. So the answer came from the study of autoantibodies. Patients with autoantibodies, once they accumulate 2 or more of such autoantibodies, they have 100% lifetime risk of type 1 diabetes, clinical type 1 diabetes. So that enabled our work to try to intercept early, and we leveraged the properties, the mechanism of action of teplizumab to deactivate the autoreactive cells to try to do a non-chronic T cell modulation. And luckily, the early studies were successful with a 2-week course of teplizumab. We achieved a 2-year median delay of the clinical disease. The challenge then was to talk to regulators about this new paradigm, because regulators are used to treating disease. They are not used to preventing autoimmune disease. So we had to design the right studies. We had to convince them with the right endpoints. Luckily, preventing clinical disease is a very robust endpoint, because if there's no clinical disease, there are no consequences of the disease, and that really carried the day. And then finally, the most difficult challenge as we transitioned into Sanofi is how to screen apparently healthy individuals for autoantibodies that indicate the disease is ongoing and that someone will benefit from an intervention despite being apparently healthy. That's why we were very fortunate that Sanofi took an interest, because Sanofi is one of the few companies that has strong leadership in immunology, metabolic disease, diabetes, and we were able to put those two together and now boost screening. That's my take-home message for this webinar. It's screening for early disease is going to enable early intervention and great clinical benefit in T1D first, but hopefully in other indications in the future.
Yeah, it's a great point, and of course, there's the value in terms of qualities, right? Quality adjusted life years that prevention can create, can be immense, but can be a challenge, as you say, both for regulators, but then for payers to think about how to manage all that. So, it's like you say, the T1D, where there's a clear progression to disease in a short time frame, right? It's maybe an easier situation to make that case, but it should extend beyond that, right? We should be able to go beyond that. Maybe that's a good segue to actually thinking about impact for patients. Maybe Mike, I'll start with you. What has been the reaction in terms of clinical benefit, right? What are patients seeing? How are physicians describing it in terms of the impact they're seeing?
Well, a couple things. Number one, I would say the speed with which Sabinco gets itch under control is really quite remarkable relative to the existing therapies. You can measure it on a graph with daily progression over 2 weeks, at which point you reach a robust, statistically significant improvement over placebo therapy. So that speed of itch relief is really something that impacts patients and physicians as well. In addition, in a lot of dermatologic diseases, the kind of specifically in the U.S., the notion of clear or almost clear as a dichotomous endpoint that is meaningful to both patients and physicians. We see higher levels of patients who achieve that bar of efficacy with existing therapies. Fewer than half of patients get to that clear or almost clear boundary, and we're exceeding more than 50% of patients getting to that threshold of clearance. So that's been very gratifying, I think, for both patients and physicians. I think we've seen in some other skin diseases that as you continue to push on the biological pathways, you can drive that ever higher. But I think kind of getting beyond the 50% mark is a great milestone for patients with this disease. So I think there's been a lot of very favorable feedback for the speed and the ultimate level of efficacy achieved with Sabinco.
Paula, maybe same question to you. I mean, again, thinking about the patient impact, the way physicians talk about it, help us. I mean, I think you already kind of oriented us to making sure that you can kind of break the compromise between safety and efficacy, but maybe I'll try to bring it to life a little bit of what does it feel like to be a patient with MS and the benefit they see when they're on treatment.
Of course. I mean, first, maybe just taking a step back, MS really comes in two varieties, if you want. There's what's called relapsing MS and primary progressive MS. Relapsing MS is mostly a disease of younger persons, that mainly females. They get relapses, so periods of acute neurological disability that usually go away with treatment, and then gradually as the years go by, you start accumulating disability, and that usually means that you get lack of vision or lack of coordination, but at a certain point your mobility really starts to be impacted. So you lose the ability to walk without a stick. Sometimes you become wheelchair bound. You lose autonomy, independence, employment. It's a very severe disease at that stage. For primary progressive patients, think of the same thing but in a more accelerated time scale. So they never really get the relapses. They go almost very, very rapidly into really severe mobility limitations. And prior to Ocrevus, relapsing patients had options. There were a few therapies, but again, there was that dichotomy between efficacy and safety, right? And primary progressive patients had no option. There were no therapies approved. And Ocrevus was really the first one. So since then, what have we seen? Well, first, we've treated over 300,000 patients globally already, which for a relatively rare disease, it's quite remarkable. And Ocrevus has become a little bit the drug of choice for most patients, both because of the fact that it's very convenient, so once every 6 months, which is very good for a young adult with an active life, right? But also because it's very safe. And even just last week, we've published a 10-year follow-up for our original patients. And what we were hoping for, which was that control of progression, meaning that how much more time do you get before you need a walking aid or you become wheelchair bound? And what we've seen really is that after 10 years, 80% of people with relapsing disease don't need a walking aid, and 70% of people with progressive disease are not wheelchair bound. And that's really remarkable. So what we hear back from patients, it's really that this drug is giving them more years of active, useful life, enjoyable life, away from disability. It's not a cure, but we get a real sense that there is a pre and post, that now there is hope, and there's something you can build on that allows people with even the more aggressive forms of this disease to have a meaningful, hope that they're going to have several years of autonomy and independence. So I'd say it's been a remarkable impact.
Yeah, that's I mean, it's incredible, right? Let me follow-up scientific question. Is there something different in the underlying biology of patients who have relapsing remitting MS versus those with primary progressive, or is it just a clinical spectrum, in the way it manifests? Do you have any insight on that to share with us?
I think it's a really interesting question because it kind of comes back to this whole discussion around B cells and T cells, right? And what we found, again, because we started looking, the data was always there, is that in more progressive patients and in older patients, what we find are these niduses, these foci of immune cells in the brains and the meninges of these patients. And these foci are actually mostly populated by B cells, by CNS resident B cells. Apparently, they become compartmentalized, autonomous, and drive the local process of gradual neuronal death, if you want. So in some ways, actually looking again retrospectively, I feel that probably the reason why ocrelizumab works so well for that type of patient is the fact that it touches the key pathophysiology that drives progression, whereas a lot of the other mechanisms really were not that direct at doing that. But again, it's serendipity and then illuminated by having a drug that actually works. Because the received wisdom even a decade ago was not true.
Yeah. Okay. Good. Super. Maybe Mike, a question for you. So Paulo and Francisco, you both talked a little bit about dosing and convenience there. I mean, I think you've all highlighted the most important thing is efficacy and safety. Along with that. But Mike, given the others have talked a little about convenience of dosing, do you want to talk a little bit about oral versus injection and how we think about, how does that matter to patients?
Yeah, that's a great question. Something we talk about a lot internally. I think it's definitely the case that with a severe enough disease, convenience doesn't matter so much. But there's always going to be patient populations for which a particular route of administration or a particular frequency is going to be optimal. People are pretty accustomed to taking pills. It's a very simple thing. People can become accustomed to taking injections, but an injection that you need to take subcutaneously once every week or once every 2 weeks, that impinges upon your quality of life. No question about it. So I think there's really been a realization that even for drugs with highly effective injectable therapies, a similarly effective oral therapy can really reach many more patients just due to the convenience. So people don't like shots, generally speaking. As I said, I think if you have a severe enough disease and that's the only way the treatment can get to you, it will be used. But the accessibility of oral therapies, I think, is something that is not going to go away. That's probably good news for our medicinal chemists that not only is it always something that patients are going to prefer to take a drug orally, but in addition, I think the technology of small molecules being able to tackle more and more targets, I think really gives a lot of job security to the medicinal chemists in this world who come up with oral therapies.
Yeah, that's great. I mean, again, I think as you highlighted, right, for patients, modality doesn't matter, right? It's the what do you get, right, in terms of efficacy, safety, and convenience, and whatever tool allows us to get there is the right answer. I'm going to shift gears on format now a little bit, and actually, rather than me asking you individual questions, try to start a group discussion amongst the three of you, outside of now talking about your product, but putting your future prognosticator hat on. Where is immunology headed? What are the big breakthroughs we have to look forward to? What are the challenges in that? And, where are you excited about the science? And maybe I'll start with Francisco's. It's obviously a very hard question, but maybe I'll start with you. And, just some discussion around, what can we expect in, let's say, the next decade? Let's not, keep our horizon too narrow. That gets you really excited scientifically, and I'd love to hear everyone in the group start to react to what each of you is saying. But maybe Francisco, you can kick us off.
Yeah. Well, I am obviously very biased, but I think in the next 10 plus years, we'll start to see disease eradication, and that will take a combination of approaches. Disease prevention by early identification of patients with disease, and it just cannot be overstated how important it is, the difference between, in the case of type 1 diabetes, the difference between parents not knowing if their kids are going to wake up next day, or having to make hundreds of decisions on diet, on exercise, etc., versus not having to do any of that and just carrying out normal lives. So disease prevention is a new paradigm that I will hope will continue to work together, working with Paulo, working with Mike, combining approaches, identifying the right nodes that are critical for each disease, and then combining approaches to try to more deeply interrupt the progression of the disease, and eventually to cure diseases, perhaps by bringing in cell-based therapies as well, to regenerate tissues that might have been damaged, and then protecting those tissues with our therapeutics. So combining prevention, better treatments, and regeneration, can we start to eradicate autoimmune diseases?
Yeah, I actually really like what Francisco is saying about eradication and prevention and cure. I mean, the reality is, for the large majority of the conditions we work in, therapies are at best modestly good, right? And so there's a huge medical need. Honestly, I wish that in neuroscience diseases were less complicated than they are. I think there's several challenges, and so trying to put on my future-looking hat for the next 10 years. I mean, the first thing that I think is going to happen is that we're going to start to be able to cross the blood-brain barrier with drugs better. So just delivering the drugs to the target, to the organ of interest, is still a major, major challenge for us in neuroscience. And therefore, I mean, there's some interesting technologies being developed now that allows us to shuttle antibodies, for example, into the brain. I think that's going to potentially make a big difference. Earlier diagnosis and treatment, I think, is going to be fundamental. And we already see that in several of the more rare neuromuscular genetic disorders. You see neonatal screening, you see like treatment after birth, essentially, with really great outcomes for these patients. And a third thing, and Francisco touched on that, I think it's really at the combination or at the interface of a lot of these modes of action that I see a lot of opportunity. And as I think about some of the big challenges for healthcare and for our societies right now, like Alzheimer's or Parkinson's, or the major neurodegenerative conditions, we know these are multifactorial, and they require interventions in multiple sites of action, basically. So take Alzheimer's, we've just seen the first approvals of new medicines, new disease-modifying medicines. Great step forward, but obviously still a small step forward. So we need to start thinking of these things not as monotherapies, but as combinations. So you'd probably need a combination of something that targets misfolded proteins, and something that targets the immune response, and something that targets the metabolic response. It's just more complicated, but I actually am very hopeful that in the next decade we'll take meaningful steps towards that.
Mike, I'd love to hear your prognostications as well, where we're headed.
Yeah, really just to build on Francisco and Paulo's comments. I thought it was interesting that Francisco mentioned cell therapy, which I'm sure he was thinking about it in terms of cells that can reproduce what the pancreas you're born with is intended to do by producing insulin in a responsive way to blood glucose. But I think one thing that we'll see in the next decade is this new story of the ability to reset the immune system with CAR T therapies that were originally designed for eliminating B cell malignancies, coming back to B cell depletion. The ability to get an ever larger population of B cells eliminated from the body. Now there's several case series that suggest that could be sufficient to reset the immune system to the point where the pathogenic autoantibodies disappear and don't come back without the need for future treatment. There have been enough cases observed to make me fairly confident in saying that's possible in some patients. Now, of course, there's safety issues with going without B cells for too long a time or taking too many of them out, but that's a viable. It appears to be a viable therapeutic strategy for at least a subset of patients who really have no other great therapeutic options. So that's one thing I think that we'll see, unfold over the next decade. Another thing, and I think Paulo touched on this a little bit as well, the ability to bring in additional orthogonal mechanisms or even additional subsets of effector immunological pathways. I alluded to this a little bit in talking about allergic or Th2 cytokines. There's actually a long list of those cytokines, and hitting 2 of them gives you a good effect. Hitting 3, 4, 5 is likely going to be even better, and we've seen that kind of play out in some other diseases. That realization that you can go back to the same well sometimes and bring more efficacy, combined with the ability to engineer proteins, for example, or to design small molecules that can be combined into a single therapy. I think we'll see that play out over the next decade. Probably the most speculative prediction I would make is that with all due respect to the importance of B cells in autoimmune diseases, I've always been a believer that the T cells are the generals of the immune system, and they tell the other soldiers where to go. So understanding the basis of the loss of tolerance in even though autoimmune disease might respond to B cell depletion, the underlying cause is almost certainly T cell driven. So advances in receptor sequencing, the ability to predict protein structure, peptide immunogenicity. I think there's a collection of technologies, including artificial intelligence, that make it likely in the next 10 years we'll have a better understanding of why people come down with type 1 diabetes to begin with. And it's probably not going to be a single, we won't think of it as a single disease, I don't think. In 10 years, we'll think of it as a collection of diseases that have very specific causes. And if Francisco is right, and we can do better as a society at screening patients and understanding who's likely to come down with autoimmune diseases, I think that creates really exciting opportunities to prevent the onset of those diseases and not have to get into a position where you're out of potential treatments and you have to bring out the big guns that inevitably will be associated with some unwanted side effects.
Yeah, so amazing set of themes. I mean, I love this last idea around, tools that are going to help us really understand the heterogeneity of the underlying etiology of things that probably right now look like single manifestations, but different diseases. A number of you made the point around combination therapies, around the hope for regeneration and or reset, with cell therapy and so on. I want to come back though to actually Francisco's point that you made a couple of times around the importance of prevention that I think you all picked up on. I mean, that if I understand the way you articulated it, right, by the time you have clinical manifestations in many of the diseases, it's too late, right? By the time you've got Parkinson's, the dopaminergic neurons of the SNA have been killed, and it's hard to bring them back, although maybe cell therapy one day can do something about that. By the time you've got type 1 diabetes, right, your beta cells of the pancreas have been killed, and it's hard to bring them back. And yet there is the promise around the immune system or immunological applications actually apply in a lot of these diseases that are really the scourges of humanity, right? I mean, think about neurodegeneration, right? There appears to be, as highlighted already, an immune component, maybe a major one. Think about, metabolic or cardiovascular disorders, atherosclerosis, right? There's obviously a big immune component to it. Even obviously in cancer, immunotherapy, right? Turning on the immune system. So can you put your prognosticator's hat on again, right? Your crystal ball, and talk more about prevention, in particular, what's necessary outside of pharma for that? Like what does the regulator need to be doing differently? How do we as a society need to be more willing to be proactive about this, as opposed to the current state, which is usually waiting until you have a disorder to treat it?
Yeah, I think the first thing is we need to understand the natural course of the disease, and as my colleagues have alluded, there are common mechanisms here in play. That trigger that Mike referred to, in many cases, it's probably an infection that breaks tolerance initially. And in MS, we have a fascinating story around Epstein-Barr virus. The same thing in other diseases. In type 1 diabetes, it appears to be an enteroviral infection, Coxsackie B virus, etc. And the same pattern emerges across most autoimmune diseases, where you have a genetic predisposition, infections that break tolerance, and then the immune system with all its different components destroying our cells. And of course, I'm oversimplifying the complexities, but in general, you can find autoantibodies years before you come up with clinical symptoms in MS, in RA, in Crohn's, in T1D, in celiac disease, etc. So can we start to introduce mass population screening programs in our societies? This is done in the Nordic countries, in regions in Europe, in Germany, in other countries. The UK is probably moving towards genetic screening for the entire population in the next 10 years. How do we gain knowledge of the predisposition to disease? How do we gain knowledge of early disease? It's going to require a societal effort. This cannot be just done by pharma. We will do our part, but we need policymakers, we need patient advocacy groups, we need society to realize that it's much better to treat early than to wait for complications.
So maybe to build on what Francisco was saying, because I share very similar thoughts. I think the challenge we face, and you mentioned a couple of the big neurodegenerative diseases, MS behaves the same way, basically, is that these are chronic diseases in which, let's say, the burden to the patient is actually the smallest part of the burden to overall society. When you see the impact that these diseases have, not just on the patient themselves, but on their family, on their caregivers, in the healthcare systems. And those costs, frankly, are not well quantified. right? Because the way that our healthcare systems and our payer systems are geared towards, it's really for acute diseases that have pretty black and white outcomes like mortality. Until, as a society, we start doing a better job of really understanding the real burden that chronic conditions, and not just in neuroscience, across in metabolics, in cardiovascular, in autoimmunity generally, the real cost that these diseases have for us as a society, we'll have a hard time making significant progress, because we need to reward investment in those areas as well. And right now in Alzheimer's, for example, we still have less than 5% of the investment that goes into cancer therapies goes to Alzheimer's. I think that's a reflection of that to some extent. I think the other thing is that, and you mentioned regulators, for a lot of these conditions that are chronic, measuring meaningful outcomes, and Francisco was mentioning like conversion to having a disease, but imagine conversion to having Alzheimer's or conversion to having Parkinson's. It can take years to do that. And therefore, prevention therapies, by definition, you want to intervene well before symptoms are set in. And of course, then you have this paradox, because you can't really run clinical trials that last a decade. It's extremely hard to have robust clinical research when your outcomes are measured in decades, not in years. I mentioned the 10 years data we've generated with Ocrevus. But MS is kind of the easy part of the spectrum in some ways. For long-term chronic conditions, that's just really hard. And therefore, I think we need a really constructive dialogue between the industry, between universities, between regulators and payers as well, to see what types of data are necessary to get approvals. Because frankly, a lot of these conditions will require that type of partnership, so that we can actually start moving towards prevention trials, which I do think it's where we get the most benefit for patients and for society as well.
Yeah, I agree with what my colleagues have said. I would say that I'm somewhat sanguine about the ability of regulators and society to step up with some of the things that are required. They're good examples of excellent diagnostic tests that aren't performed. Alzheimer's might be one example where, if there's nothing to do about a risk that you carry, knowing the answer is not particularly useful. I would point to some of the public-private partnerships that have really helped us build compelling therapeutic hypotheses. Someone mentioned the UK. The UK Biobank is a tremendous resource. I happen to sit on the executive steering committee of the FNIH, and I get to see all the projects that they are working on and have worked on over the years, which build these really invaluable data sets that drug developers can incorporate into their fund of knowledge to develop compelling hypotheses. The compelling hypotheses lead to compelling therapies, and compelling therapies lead to regulatory innovation and societal innovation. That I think, if we knew of a therapy that would prevent diabetes from developing in patients who've begun to develop autoantibodies, I have no doubt that we would find a way to institute screening programs and
the proof is in front of us, right? So
yes.
And I think that has been true, actually. Sorry, that has been true, actually, Mike, in rare neuromuscular, right? For example, spinal muscular atrophy, where now neonatal screening is basically commonplace in the US and becoming very commonplace in other parts of the world as well. But it's because we have available therapies.
Yep.
So I know I promised some time for audience questions. So if you should have a way to post them through to us, although I'm not seeing any, I'm probably not looking in the right place, but please post your questions if you have any for our panelists. Just make sure I'm looking in the right place. It would be on the Q&A tab. I think if you hit your Q&A tab, you can post a question there. Okay, it looks like we don't have any yet. I'll leave that open. So, as anyone wants to post a question, we can definitely field it. But if not, what I'll do is just, turn to wrap up. First of all, just a huge thank you. I know, Mike, Paulo, Francisco, how busy you all are, doing not just obviously work on these drugs, but on the next round of discovery is going to help patients all around the world. So taking time out of your calendars to share your wisdom with us is incredibly appreciated. Thank also to our audience for joining us. The ability to share this knowledge, I think, is gratifying to the panelists, right? So it's important that we all are in this together. And lastly, thank you to Galien for hosting all of this. The webcast is available if you go to the Galien Foundation homepage, which is just galienfoundation, all one word, .org, as well as all the other materials regarding not just this session, but all the other sessions. A reminder that we have the Thursday forum in New York that begins at the Alexandria Center for Life Sciences at 7:30 in the morning. That has keynote from pharma CEOs from the US and Europe, and concludes with the presentation of the 2023 Roy Vagelos Pro Bono Humanum Award winner, followed by the awards gala. Both of those are held in the evening at the American Museum of Natural History, also in New York. So if you're able to join us for either of those, it's a truly fantastic experience, deep in science, and also appreciation for what science is able to do for the human condition. So thank you all. And I'll just double check that there are no questions for you. So we can give everyone 3 minutes back, and appreciate you attending and participating in this.
Thank you.
Thanks, everyone.
Thank you.